Hello everyone. Once again I welcome you all to my series of lecture, that is Understanding Pharmaceutical Science with Dr. Hariharan. So today we are going to discuss about the production of polio vaccine. So polio vaccine production is a very important criteria because it is one of the alarming disease which should be needed to be eradicated. So this discovery of polio vaccine has given an immense understanding about the infection process also. So first we understanding about the polio, that is the poliomyelitis. So poliomyelitis is one of the much feared disease. The reason is that once this polio virus gets infected, the patient has a chance to get paralyzed because this virus generally causes an infection to the central nervous system, thereby it causes the paralysis. So if you see the polio infection, not all the virus is going to cause the paralysis. Generally, often if you see that certain viral infection of this polio leads with a mild and non-paralytic forms, it can just go away with the stimulation of the immunity itself. If you see that 90% of the infection causes no symptoms, the person started getting immunity against that virus naturally, whereas the 1% of the person are get paralyzed. So to save this 1% of the population which is generally going to paralyze and going to suffer to their entire life, so the people started getting vaccinated with this polio vaccine. So if you understand the infection process, how this poliomyelitis virus works is that generally this virus invades our body through the oropharyngeal route. So it goes through the mouth, then it passes to the stomach and gets colonized, gets internalized with the mucosal cells, that is the intestinal mucosal cells. So this virus gets infected the intestinal mucosal cells. So by this infection, what happened is that our body started producing antibody against it and develop its own immune system. So what happened is that these cells get replaced by the mucosal membrane by the cell replacement process, and it stimulates the long lasting immunity also. Whereas what happens is a small amount of the infected virus will bypass this intestinal replacement of cells, then it enters into the blood through the lymphatic system. So once it comes into the blood, it crosses the blood-brain barrier and reaches the central nervous system and started degenerating the nerves. So this degeneration of nerves will lead to the paralysis. So to overcome this, we have to inhibit the virus to pass through this bloodstream into the CNS. So that was the full core objective of development of this vaccine, whether it has an inactivated form or if it is in an attenuated form. So if you see the picturized representation, what happened is that the infection process, the person gets inhaled or the person gets consumed through foods, it goes through the oropharyngeal route and gets infected at the mucosal surface of the small intestine, that is the intestinal mucosa. Then it passes through the lymph nodes, then finally into the blood. From the blood it reaches to the central nervous system, thereby it started the degeneration of the neurons. So because of this degeneration only that leads to paralysis. So while thinking about the polio vaccine to protect the peoples against this polio, there are two different types of polio vaccine was developed. So the first one is the inactivated polio vaccine, otherwise we can call it as a killed polio vaccine. It is also called a Salk vaccine. So once to honor the American virologist who developed this vaccine. And second is the attenuated vaccine, which is also called a Sabin vaccine because it was developed by Albert Sabin. So these two vaccines has revolutionized this polio vaccination program, which has been contributed by many countries to eradicate this disease. So let us understand the production process of both the vaccines. The first one is the inactivated polio vaccine. So it was developed by Jonas Salk, as you know, that he was one of the reputed American virologists to develop this vaccine. So that's why also called a Salk vaccine. So if you see this inactivated polio vaccine, it is administered prophylactically through parenteral route. So whereas the second type, attenuated vaccine, is given orally. So this inactivated polio vaccine is given parenterally. So by parenterally administering this killed vaccine directly into the bloodstream, what happens is this viral particle will stimulate the immune system for the production of antibodies in the blood. So what happens is, once the person comes in contact with the real viral vaccine, so his body contains the enough antibodies in the blood. So if the virus escapes from the intestinal mucosa and gets into the blood, these viral particles will neutralize it and gives protection to the person against this, because once the virus reaches the blood circulation, then only it can reach to the CNS. So the presence of these antibodies in the bloodstream neutralizes the virus, thereby it offers the protection to the person. So however, this virus is not going to provide immunity through the natural infective process of the virus. That's why how the virus goes through the oropharyngeal route into the intestinal mucosa. So it does not do is, only it produces enough antibodies only at the location of the bloodstream. So thereby it provides the protection. So generally what happens with this vaccine is that if a person is administered with inactivated viral vaccine, the person may be prone to infect other peoples, because he becomes literally a carrier of the viral load. So once he comes in contact with other person who is not vaccinated, so he can infect that person also. That's the only drawback with this vaccine is having. So if you see the preparation of this inactivated viral vaccine, so I have already told that it is also called a Salk type vaccine, because it has named after the invention by the American virologist who actually developed this vaccine. So this viral vaccine is produced in such a manner that first the viral is get cultivated. So generally it is cultivated in a monkey kidney cells, then it is harvested. So after harvesting this viral suspension, it was confirmed that first to check one, whether it carries the correct strain of poliomyelitis virus, because poliomyelitis carries different strain, but the most preferable strain which we need for the vaccine production is the poliomyelitis type 1, type 2 and type 3. So the first one is after cultivation of the viral suspension, we have to check whether we have cultivated the correct strain of the polio virus and it is present in the suspension one. Second, we have to check whether the viral titer value is enough, so that can be administered as a vaccine. That means we need a minimum level of viral titer value. Third, then it should be free from other viral microbial, that is the bacterial and the fungal contamination. So this viral suspension is used for the next step. That is, the next step is the inactivation step. So in inactivation, what we are doing is we are adding a 0.01% formaldehyde solution. So this formaldehyde causes neutralization, that is the inactivation of these viral cells. Once it is stirred with the help of a magnetic stirrer under a controlled condition, that between an optimum pH and temperature, if it is stirred continuously, the formaldehyde causes neutralization of this viral particle. So it does inactivation process that can be completed within 6 days, but anyhow we will continue the process for 12 days. Otherwise, that means twice that of the 6 days, that is the 12 days. Otherwise, we will continue until there is no virus remains in the preparation. So the suspension is filtered every halfway point. Then it is checked for the absence of the infective virus. So generally, we will draw the sample at the 9th day and the 12th day to check the absence of this infective virus. So once there is no virus is there, infective virus is present, then we got that univalent vaccine from every viral suspension. This univalent vaccine is that is the univalent preparation. That means univalent means it has an ability to stimulate the antibodies against a particular epitope of the polio virus. So each virus, that is the type 1 can produce a particular epitope, so it can produce a particular antibody. So type 2 can produce it. So these univalent viruses are then blended, that is it is mixed to produce a trivalent product. So then it is further tested the absence of the infective virus. Then finally the formaldehyde, which might be somehow traces will be remaining in the preparation, should be neutralized. So the formaldehyde is neutralized with sodium metabisulfate, and finally the preparation is kept in presence of a bactericidal agent, that is thiomersal. So the problem with thiomersal is that if any heavy metal is present in the vaccine is that it interacts, then it can cause us toxicity to the viral vaccine. So to avoid this one, to maintain the thiomersal in a perfect condition, we will add disodium EDTA into the preparation. So this disodium EDTA forms sesquester with the heavy metal and prevents the reaction with the thiomersal. So the decomposition of thiomersal doesn't happen. So because of this, the viral will be protected, vaccine will be protected, and this is administered as such as a parenterally for the protection against the infection. So the second type is called the attenuated polio vaccine. So it is also called a Sabin vaccine because it was developed by Albert Bruce Sabin. So this attenuated vaccine is more or less work similar to that of the infection process. So the attenuated vaccine is administered orally in drops. So that's why we currently administer most as an attenuated vaccine to the children. We give 2 drops. So what happens is that by the oral route, which is the most preferred route, so it stimulates the immunity. So what happens is that this goes into the mucosal layer in the alimentary canal. So once it goes, it stimulates the immunity, and preferably how it stimulates the immunity is that it binds with the mucosal cell, releases the interferons. So this interferon stimulates the production of antibodies by the lymphocytes. So once the original viral, that is the infective virus, enters, already the ability of the individual to produce an immunity against that virus, it will inhibit it. It prevents the establishment of the infective virus in the mucosal layer. So if you see, the principle of the attenuation process is based on the idea that there are 3 distinct antigenic types are available for the polio virus. That is already I told you, polio 1, type 2 and type 3. So we need a vaccine which is stable, which will be polyvalent in the nature with these 3 characteristics, so that it can give a protection from other types. So later on, after understanding of greater antigenic study of this type 1, type 2 and type 3, it was identified that if we give a vaccine for one, it itself started giving protection from other types also. So therefore we want to include one important strain, so which can produce a polyvalent vaccine that is satisfactory enough to produce a long lasting immunity. So this was the ideology of the production of this vaccine. So we take a virus, particular strain, which can give you a broad spectrum of activity. We take that strain separately, and it is cultivated in the rhesus monkey kidney cells. So in a fixed cell culture of the rhesus monkey kidney cells, it is infected and gets cultivated. So preferably we know that the polio vaccine, that is the poliomyelitis virus, generally infects nerve cells. That means it has an ability to grow in the nerve cells. So what's the problem? We can cultivate in a nerve cells also, but the problem with the nerve cells is that it has a short of life, one thing, and the second thing, if that any traces of proteins of nerves comes in the vaccine, it can cause allergic reaction to the brain in the human cells. So that's why we avoid this nerve cell and we choose the rhesus monkey kidney cells. So before the cultivation of this viral into the rhesus monkey kidney cells, so first the rhesus monkey kidneys rhesus monkey used for cultivation is quarantined and checked for any tuberculosis infection is there or not, or any other communicable disease is present or not. It is checked even before and after the death also. Then the kidney cells is continuously propagated. This cannot be propagated for a long time, because what happens, this kidney cells has an ability to convert to a malignant cell lines and then the normal cell lines, and it can causes a problem to the peoples. So how we are preparing this attenuated oral vaccine? So since I already told, it is also called a Sabin type vaccine, because the person Albert Sabin who has developed it. So what we will do is, we will cultivate in the same manner through the rhesus monkey kidney cells, what we have done in the Salk type also. It is produced in the similar manner, that is, inoculate the virus, grow in the kidney cell until the proper amount we get it. So whereas in the Salk type and this type, what the difference exists is that the attenuated strain in this case is prepared by continuous passage, that is the continuous subculturing into the monkey kidney cells, whereas in that case we need one cultivation until the enough titer value is reached, that is enough for that the Salk type. Whereas in the Sabin type we have to go for the rapid passage into the kidney cells, that's the one. And in the second, the virus in the final vaccine may not represent more than 3 subcultures of a laboratory. In this type, what happened is that in the Sabin type we will check whether there is a presence of any active virus is present or not, whereas in this type what we will do is, before we pool it and make a final vaccine, so we get the 3 subcultures from 3 different laboratories from a laboratory, and the clinical test should be done until the satisfactory is achieved, that is, it is attenuated and no living strain is present, that should be checked. Where in that case we have to check only for the living strain. In this case there is no inactivation stage. In that case, in the Salk vaccine we inactivate with the help of a formaldehyde. So after preparation we will generally check for the presence of bacteria, molds, fungus or viruses. Then a special test is carried out to check there is any virulent strain is there or not, because after attenuation if a single virulent strain is there, that also problematic for us. So this is all about the production of the both the killed vaccine as well as the attenuated vaccine. So if you see the dosage and administration of this, so for the children's it is generally preferred, the polio vaccination is generally preferred in children rather than the adults, because adults we have certain problems. For children's it is given in 2 types. One is we give a primary series of vaccine, we give 3 doses, and we also give the booster dose for the lifelong protection against this virus. So the primary series is generally a 3 dose of comprising about 0.5 ml. It can be administered intramuscularly or subcutaneously if it is a Salk vaccine, or given orally in case of a Sabin vaccine. So it is preferred at the 2nd month, then the 2nd dose at the 4th month, and the 3rd dose between 6 to 18 months. So in no circumstances this vaccine should be given more frequently. So if you give more frequently, then it causes some undesirable reaction. So generally a gap of 4 weeks is needed between 2 vaccination. So the first immunization is over within the 18 months, then the booster dose can be given at different interval of time up to 4 to 6 years of the age. So after that there is no need for administration of this vaccine. So if the vaccine is not given during the childhood, if you have to administer for the adults, then means if you see the dosage regimen of this unvaccinated adults, the primary series is recommended for unvaccinated adults in such a manner that a care should be taken, because by the administration of this primary series, that is the 3 doses, sometimes can cause a risk the person gets exposed to the polio virus. naturally. So generally this is given once there is a, generally the unvaccinated adults is not preferable. Generally we prefer to give once it's getting into a area where the polio virus may be in abundance, that means he may exposed to that polio virus, then only we have to administer it, otherwise itself it is a problematic one. So what happened is that the dosage recommendation is that 0.5 ml is given as a first dose, then after a gap of 1 to 2 months the second dose is given, then after a gap of 6 to 12 months then the third dose is given. That is more than enough to provide an immunity for the unvaccinated individual. So if you see the storage condition, the storage is similar to that of the other vaccine, that is generally 2 to 8 degree is preferred for the storage condition, that is 35 to 46 degree Fahrenheit. Then it should not be freeze and it also should be protected from the light. So thank you very much for understanding about the basic production process about the both the live as well as attenuated polio vaccine. So thank you very much. Continuously watch my other lectures also. Bye.